on to the final speaker of this session, and this is Giulia Schiroli. And she will be talking about highly efficient correction of the sickle cell disease mutation in patient HSCs using an RNA gene writing system and RNA-based nuclease-free approach to genome editing.
A bit long title. Thank you so much. I'm delighted to be here on behalf of the Tessera team to present our progress towards sickle cell correction in hematopoietic stem cells using RNA gene writing system. So at Tessera, our science is based on re-engineering mobile genetic elements. So basically leveraging what nature has already evolved in order to generate novel genetic engineering tools. So mobile genetic elements encode for all the machinery necessary to move themselves from a site in the genome to a novel site. And they do it through different mechanisms. Some of them use DNA as substrate, and some others use, such as non-LTR retrotransposon, use an intermediate of RNA where transcription happens and then reverse transcription. And then this enables the integration of the cDNA. So at Tessera, we engineer both of these platforms, but for today's talk, we will focus on RNA gene writing system, since this platform is the most amenable for non-viral in vivo delivery. So basically, our engineered transposons that we call RNA gene writers, they have multiple functional domains. Which are engineerable, and they utilize as a mechanism of action target prime reverse transcription. So basically, target prime reverse transcription starts within the nuclease domain, nicking the host DNA, which is then followed by the priming of the 3' end of the RNA template, which is then starts the reverse transcription, followed by the integration of the complete DNA sequence. So the approach that we took at Tessera is to high throughput screen more than 100,000 of combination of different protein domains and cognate RNA templates in human cells in order to be able to generate different classes of RNA gene writers. So each of them has a different biochemistry, and they are able to introduce small point mutation, or some other platforms are even able to integrate much larger transgene cassettes. And I highly invite you to the talk of Giulia tomorrow to have a much in-depth presentation of all the different type of RNA gene writing system that Tessera is developing. So all of these platforms are fully compatible with all RNA delivery when coupled with an appropriate delivery vehicle. So for this presentation today, we will focus on the platform that enables to introduce precise genetic modification at any site of interest. And this platform enables basically to virtually introduce any kind of mutation, being a substitution, an insertion, and a deletion. We decided to apply this platform for sickle cell disease. So sickle cell disease, as all of you know, is due to mutations in the a specific point mutation in the HBB gene, which leads to red blood cell sickling and multi-organ complications. So the recent advances in gene editing and gene therapy has demonstrated, importantly, substantial clinical benefit from bone marrow transplantation, with the idea that if we correct long-term multilineage stem cells, this treatment can provide a lifelong correction for sickle cell disease. However, they have also highlighted the limitations of this approach, and those limitations are mostly related to toxicity of the bone marrow transplantation procedure, cost, and importantly, patient outreach. So the approach that we want to take at Tessera is to try to develop an in vivo gene correction strategy for sickle cell disease by exploiting our RNA gene writing system in order to revert the sickled hemoglobin to the wild type 1 by correcting the mutation. So in this case, as a reminder, our gene writing technology is composed by different RNAs, one encoding for the gene writer itself, which is in this case a Cas nickase fused to a retrotranscriptase domain, and a cognate template RNA, which encodes for the modification that we want to introduce. So we just developed and optimized the technology to be able to efficiently genetically engineering hematopoietic stem cells, and so we deliver either by electroporation of oral MP delivery these RNA in healthy mobilized human peripheral blood stem cells. So in this case, since we want to model the correction in wild-type cells, we are installing a benign Makassar mutation just for the sake of monitoring the editing that we want to introduce. So by performing different rounds of optimization of our both the gene writer and the template itself, we were able to obtain a very high efficiency of what we call a perfect rewriting, which means the introduction of the specific base pair change that we want to introduce without any unintended edits. And as you will see, these 80% of allele modified almost represents 100% correction of all the cells. We also obtain very good results by LNP delivery of our technology. We then went on and demonstrated that our editing efficiency occurs at very high fractions and at very same efficiency in HSC-enriched and progenitors-enriched populations by utilizing a classical surface marker to be able to discriminate those populations. And we also took a step further and demonstrated that for our editing process to occur, cells don't need to be actively cycling. In fact, we can obtain a similarly high efficiency of editing either if we culture the cells for 48 hours in cytokine-rich media, as normally has been done for gene editing or gene therapy application, or if we don't pre-stimulate the cells at all, rendering our platform very suitable for in vivo delivery approach. We also characterize the toxicity of our platform in hematopoietic stem cells. So in this case, we are comparing our RNA gene writer to a wild-type Cas9, which is introducing a double-strand break. As a reminder, the TPRT mechanism doesn't introduce a full break, but only a single nick. And this double-strand break has been characterized to be very detrimental, especially in quiescent HSC from the literature. So by performing RNA-seq analysis, we were able to observe in the wild-type Cas9 treatment upregulation of pathways already characterized by the literature as consequence of the double-strand break, such as upregulation of P53 and downregulation of cell cycle-related pathways. None of this alteration was observed in a significant way by our technology. And we also try to characterize more in depth this DNA damage response mediated by the nucleases, scoring our samples for a signature which was already characterized by the Naldini lab as a signature of DNA damage response induced by a single break in hematopoietic stem cells. And as you can see, this signature is highly upregulated in a wild-type Cas9 cutter, but we observe absolutely no upregulation of these in our rewriting sample. This was also nicely associated with a reduction in apoptosis, which is a known consequence of double-strand break in hematopoietic stem cells. We then went on and validated the long-term reconstitution potential of our treated cells by performing xenotransplantations into hematochimeric mice. This is a pool of 2 independent experiments. As you can see, we did observe no difference in engraftment levels measured over time and long-term in the bone marrow. And nicely, we were able to confirm high and stable levels of editing from in vitro to in vivo, and then in bone marrow in lineage-negative CD34 positive cells and all their multilineage progeny, thus demonstrating that our technology doesn't influence HSC long-term self-renewal. We also plated CFU assays out of the lineage negative CD34 positive cells at long term from the mice in order to measure the fraction of cells that we correct with our technology. And basically, we were able to observe that we are editing more than 90% of the CFU progeny, with more than 60% of them bearing biallelic editing. Thanks to a fantastic collaboration with the Boston Children's Hospital, we also were fortunate to have access to sickle cell disease cells. In this case, we are not introducing anymore the Makassar variants, but we are actually correcting the sickle mutation back to wild type. Even in this case, we were able to observe high levels of editing efficiency in the CD34 positive cells, which was importantly maintained also after erythroid differentiation of the treated cells. No skewing in the total number or type of colonies was observed upon our treatment, and again, by performing allelic modification count on the single CFUs, we were able to confirm that with our editing technology, we are able almost to correct all the cells. We then went on and tried to validate the functionality of our corrected cells by performing differentiation into RBC assays. By performing erythroid differentiation, we were able to see that our edited cells have actually an advantage and produce more mature cells as compared to sickle untreated one, sorry. By performing molecular characterization of the levels of HBS and HBA, we were able to observe that we are actually able to restore 98% of physiologic levels of HBA, completely abrogating the production of the HBS. These results was also recapitulated by performing sickling assay in under hypoxic condition, where our corrected red blood cells show complete absence of sickling. So finally, in an attempt to bridge our editing technology to in vivo editing, we are also showing data regarding in vivo delivery into hematopoietic stem cells in mice and NHP. So as a first step, we are delivering lipid nanoparticles encoding for a GFP mRNA. So we first perform these experiments into humanized mice that we treat with a single dose of lipid nanoparticles, and 16 hours later we collect the bone marrow and analyze different populations. So here we are reporting that by optimizing different formulation and experimental parameters, we are able to see functional GFP expression in more than 80% of lineage negative, 34 positive and 38 negative cells, so quite a pure HSC population in these mice. And nicely, these results were also recapitulated when moving into larger animals. So with a single dose of LNP, we can deliver functional GFP expression in up to 30% of HSPC. So overall, I hope I convinced you that our RNA gene writing technology can be used to efficiently engineer primary HSC, and in particular to correct the sickle cell mutation to wild type, that our HSC can maintain their hematopoietic potential as measured by different assays in vitro and in vivo, that we are able to correct with our technology patient-derived HSPC by reverting the sickle mutation back to wild type, and thus our corrected cells were indeed expressing almost 98% of normal HBA levels. Finally, given our ability to deliver RNA to HSPC in mice and monkeys, we believe that our strategy provides a promising new therapeutic approach for in vivo correction of sickle cell. So let me conclude by acknowledging all the people that are contributing to this project, all the contributors from Tessera and our external collaborator, Jean-Antoine Ribel and John Manis, for their great help, and happy to take any questions.
Very nice presentation. This presentation is now open for questions.
Hi, Giulia, beautiful data. So one of the questions is more technical. So when the retrotranscription occurs, so you have the tethering towards your site, okay, of the RNA. and so then the retrotranscription occurs, or is occurring also already, very early at the beginning?
No,
you need to be in the nucleus.
The retrotranscription occurs when the RNA is tethered to our predetermined genomic site.
Oh, so is it,
Yes.
is it sequential?
Yes.
So that could actually increase the specificity, you think? So let's
I
say
mean,
you reduce the off-target integration somewhere, the transposition somewhere else.
I mean, yes, we are modified the template in a way that it's tethered to the site in order to actually have a very good level of specificity.
Yeah, very cool. Thank you.
Thank you.
Yeah, maybe a quick question from my side. So how can you comment on the versatility of the approach? How easy is it to sort of define a new target site and to engineer your tool to find that and basically make it working?
I mean, it depends on the application. If you want to engineer small base pair changes with the pipelines and the optimized reagents that we have developed, we can engineer with high frequency, a lot of different sites. We had different presentations presenting our improvements towards different sizes. It becomes a little bit more difficult when you have a bigger insert. And for that, we believe that different classes of our transposons can behave in a different way, and so we might choose one versus the other.
Okay, thank you.
It's a very nice talk. Thanks. I'm from Insoma, so I just wonder a technical question. So you actually simply combine this Cas9 or Cas. It's a nickase or it's a double wild type Cas?
Yes, we have a Cas nickase fused to a retrotranscriptase domain.
And fused to reverse transcriptase.
Yes, correct.
And then, so your iron template is actually, it's not fused to your whole construct.
We have different configurations. In this particular case, it's not fused.
Okay, so you use regular guide RNA as a
It's a modified template RNA,
yeah.
so it's...
what's the advantage of, compare with prime editing?
I mean, they are both, I think they are complementary approaches. They are both based on the mechanism of TBRT. So the mechanism of action is very similar.
Yeah.
I'm not sure if there is a real advantage, but this is our, we are very confident that the technology that we are developing, it's very specific, and it will allow us to obtain high levels of editing in vivo.
Good. Thank you.
Mike Treebwasser, University of Michigan. Congratulations on an amazing study. A follow-up question to that is, have you looked at off-target site writing or genome-wide?
We haven't looked for this target yet. We have some preliminary data in other targets, such as PKU that was presented, and the platform really looks specific thanks to the multiple components that are present in our template. This really increases the specificity as compared to a wild-type Cas9 itself. Of course, it's something that we'll have to look also for this program, but we haven't yet.
Okay, we do have one online question from Eric saying, is the LNP used selected to have HSC selectivity, specificity?
We are testing different LNP configurations. We are seeing that some home to the marrow preferentially as compared to others, but we are not giving away more details on this.
Okay, and I guess there's one more online question. What was the % of off-target RNA writing? Are there indels? I think that might have
we haven't analyzed, as I was saying, the percentage of target. We do see some indels, which results probably from some residual DNA double-strand break induction that you might have. As I showed from the CFU assays, we have a very high levels of rewriting activity, so the perfect genetic modification that we want to introduce. And then secondly, even if we have indels at this site, we are actually not going to harm since we are just correcting, we are just basically converting a sickle allele into a possibly thalassaemic one if we are unlucky. So it's could be they are present, but it's not a really big concern for therapeutic efficacy.
Very much.